Pronto, estamos no ar. Rafaela.
Hello. Hello, boa tarde a todos, a todas. Estamos aqui hoje, né, dia 17 de outubro, para comemorar, para falar sobre o International Sawfish Day, que é o dia dessa espécie muito importante aqui para a gente, uma espécie de raia, né? E nós hoje temos um convidado especial, que é o James, que ele já tem uma parceria aqui com o laboratório, mas antes, deixa eu me apresentar, que eu comecei a falar e não me apresentei. Para quem não me conhece, eu sou Rafaela Brito, sou colaboradora do laboratório do Grupo de Estudos de Elasmobrancas do Maranhão. A gente atua aqui na região, na zona, na parte continental também, porque também tem raias de água doce. Mais recentemente, nós começamos a comemorar o International Sawfish Day desde 2018. A gente tem feito essas comemorações, a gente faz ações de educação ambiental em diversas áreas, tanto no meio acadêmico, como também de forma mais ampla para acessar toda a população. A gente fez, 2 anos atrás, a gente fez num shopping, né? A gente fez uma interação bem legal, com exposição de imagens, com interação com crianças. E do ano passado para cá, devido ao cenário atual, que todo mundo já está mais do que acostumado, a gente está fazendo as lives para conversar e para não deixar passar em branco, né? Então, hoje a gente tem o James, que já trabalha aqui na nossa região há um bocadinho de tempo, tem alguma experiência aqui para compartilhar com a gente sobre essa espécie que é muito ameaçada de extinção, infelizmente, e tem sido bem restrita aqui à nossa região costeira, né, amazônica. Então, James, hello, fique à vontade.
Hello.
Se apresentar. Tudo bom?
Sim, tudo. Sim. Muito obrigado pela...
Eu dei uma olhada. Vou falar.
Então, yeah, thank you, everyone. I'm going to speak in English because my Portuguese is really poor right now. But thank you so much for having me for International Sawfish Day. And I wanted to share with you just a small amount of what we've been doing using chemistry in the rostral teeth of sawfish to understand what they do and where they go. Part of the problem in conserving sawfish is that there are so few sawfish and they're so very hard to track that understanding what habitats they need and where they move is something we don't understand very well. And it's also very difficult to get that information from the few sawfish that are out there in the world. And one way that we might be able to learn this information is through records that are kept in the bodies of sawfish themselves. And I'm just going to share my screen and show a little bit of data and kind of talk about what I do. So let me find the right screen. Okay. Okay. So sawfish has these amazing rostrum with teeth that come out of both sides, and those teeth are really pretty cool. There's a lot that we can learn from them, I think. Part of what I do is use the chemistry that's stored in the hard parts of fish to reconstruct their migration. And I've done this a lot with salmon. You can use the otolith. It's a small hard part inside their head, underneath their brain. that grows like a tree, and every day it adds a new layer. And in those layers is the chemistry related to the water that fish is in, and so you can retrace the migration of a salmon using its otolith. But sawfish don't have otoliths. They're a cartilaginous fish. Their skeleton is made of cartilage, and the analogous piece that they do have in their head doesn't continue growing through the life of the fish, so you lose a lot of information. You can use their vertebrae, but that requires killing the fish, and most people don't save those. And of course, we don't want to kill sawfish, so finding new ways to get that chemical information is important. This project started, it would have been 3 years ago, I guess. I realized that my university here in Idaho has 2 sawfish saws, or katanas. There's an adult and an embryo rostrum in the university's collection. And I wanted to know if you could use the teeth from those saws to understand whether those fish were moving in and out of salt water, whether we could tell anything about the lives that they led. And the reason that I think the reason that I thought that there would be chemistry, interesting chemistry in those teeth, is that each of those teeth grows through the life of the fish. As the fish grows, it's like a pile of cups. The first part of the tooth is like one cup stacked upside down, and as it grows, another layer forms and it pushes the tooth up like this, right? And each of those layers might have chemistry related to where the fish was born, but no one had ever studied this. And I had been talking with Tommaso Giarizo from Pará, who had been talking with George. We had already met once in 2017, and we thought that this was possible. But we didn't have samples that I could get my hands on until I realized that we had these samples in our collection at my university. So I got permission to analyze these samples and did most of this work, I should say, with an undergraduate of mine, Brianna Graves, who really deserves a lot of credit for the preparation of these samples and kind of learning how we could do this work, because this was the first time that anyone had ever tried it. And she did a huge amount of work as an undergraduate student to make this happen. So no one had ever tried analyzing sawfish teeth, and when we tried, we did various things to try and understand what was going on. We tried to look for growth rings in those teeth. We looked at staining and cutting them in different ways. We didn't find anything particularly strong information about growth. You can see, it looks like maybe I'll have to go out of this presentation and zoom in. I'd like to see this a little bit better. So these are the pictures of the teeth, and the embryo is on the right. You can hopefully see my cursor, and then the adult rostral tooth is on the left. It's been cut in half because we didn't have enough space in the machine to analyze it all at once. You can see, especially down near the base, those stacking cups. of the different layers as it's growing. So on this side is where it attaches in the rostrum of the fish. So that's the youngest part of the tooth. That's telling us the chemistry of what's happening right now in the life of the fish. And as we move this way, it's a record of what has happened in the past to this fish until you get to the very end. And that is the last record that we have of what is happening in the life of the fish. So that kind of gives you some way of understanding the data that I'm going to present. When we did this, we analyzed it for multiple types of chemistry. We analyzed for strontium isotope chemistry, which is the ratio of strontium-87 to strontium-86. That is really useful, especially in freshwater fish. In saltwater, it's less useful, but sawfish, especially large-toothed sawfish, move in and out between freshwater and brackish water and saltwater. And so we can, there's a, in this plot here, you can see there's this blue line. The ocean everywhere in the world has the exact same strontium isotope signature, and freshwater looks different. So when the signature of the sawfish tooth goes to, when it meets up with that blue line, that indicates that the fish is in salt water and not in freshwater. So when we're looking at this graph, we'll talk about the adult mostly. At the beginning of its life, that strontium isotope signature is very different from the signature that we would expect to see in the ocean. So this fish is not in salt water, according to the strontium isotope signature. But then as it grows, the signature gets closer and closer and closer to 70918, 0.70918, which is the global marine signature. It never quite hits, and it's important to understand that this strontium isotope signature, we have 3 digits here, but it's accurate down to the 5th digit. So the line really has to touch that blue line to be ocean water or be very, very close. So right here, it's very, very close to ocean water, but then it starts to move again. And towards, so this is moving towards oh, I'm sorry, I'm moving the wrong way. So it starts life on the left. I'm sorry. So it starts life over here outside of fresh water, and it moves to more oceanic water, and then it moves away again. And when this fish was caught, it was captured in Belize in 1968, I believe. We don't really know where, and we don't really know who caught it, but we know that it was captured near shore in Belize somewhere. It was clearly not in ocean water. Now, on the other hand, the embryo has a very stable signature. Now, it's living inside the mother. It's not been born yet. So its signature reflects whatever the mother's signature was. And that signature may change very slowly over time, and especially as it's, that chemistry has to transfer to the young fish, and that fish is living off of the yolk from the egg for some of that time. So we don't really know how that changes, but we do know that this signature looks a whole lot like the ocean. Then we also looked at 4 other signatures. We looked at the ratio of strontium to calcium, the ratio of barium to calcium, and we're using calcium because the tooth is made mostly of calcium. So by creating that ratio, we can we can ground truth our numbers, essentially. We looked at magnesium and manganese. So all of these are under some sort of biological control. The body doesn't know what to do with the different isotopes of strontium, so what is in the water is in the fish. But in terms of these elemental ratios, the body is changing them. So sometimes these are affected more by temperature or by growth rate. But strontium and barium to calcium especially tend to reflect the water that the fish is in. And we would expect very high strontium to calcium ratio when a fish is in the ocean. So there's another story to be told in strontium here. As the fish is aging, the strontium to calcium is pretty low. This is in the adult. And it peaks a little bit right at the same spot that the fish looks like it's in the ocean here, but not very much. And then it goes down again, and then it's flat, and then there's some noise over here as you get to the end of the tooth. But it's not spiking up like we would expect if the fish was in salt water. I would expect it to be up in this range, and it's not. So that indicates that this fish never really went to the ocean. It wasn't in open ocean water. It was in brackish water that was salty and had a lot of influence from the ocean, but it wasn't pure ocean water, which is kind of what we would expect for a large-toothed sawfish. They're not a pelagic ocean species most of the time. They're in brackish water. Interestingly, the embryo has very high strontium, and that matches quite well with the strontium isotope signature that looks very much like an ocean signature. It does dip down here, so that would indicate that as it was gestating inside the mother, it started out at a signature that was a little bit more brackish, and then moved up to be much more oceanic. That's weird because we don't expect the mother to be spending a lot of time in deep ocean water. So I can't really explain that other than what we know. We don't know anything about where this fish came from, this embryo. Yeah, it's a mystery because we wouldn't expect a large-toothed sawfish to be in the ocean, but this would tell us that it is. Now, barium tells us another story. So this red line here, barium works opposite of strontium. So in the open ocean, strontium is high, and in freshwater, strontium is low. Barium is opposite. In freshwater, we expect higher values of barium, and in the open ocean, we expect almost none. As soon as water with barium in it touches salt water, there's a chemical reaction that actually creates insoluble salts that fall out of the water. So as soon as fresh water hits the ocean, all the barium disappears, and there's very, very little barium in the ocean. So, based on what we know from the strontium isotope ratio and the strontium to calcium ratio, we would expect the barium calcium ratio to be opposite of what we saw in strontium, and that is what we see. We see high values of barium early in life when the fish was far from ocean water. And then as it starts to move to look more like the ocean, the barium starts to decrease. So that's another piece of evidence indicating that this fish was in brackish water, and it started water in much more fresh water. and it moved into water that was more and more salty for the middle of its life. And then we start seeing the barium spike up again, just as we see the strontium changing back to something that looks more like fresh water. So that tells us that, and it's just another piece of information indicating that this fish moved into fresh water before it died. And again, the embryo looks a lot like ocean water. There's almost no barium in this sample. It's extremely low, way lower than the barium in this sample. So that again indicates that the embryo was in a mother whose chemistry reflected the ocean very strongly, as if she had been swimming around in open ocean salty water for quite a long time in order to build up that chemistry in her body. So that's fascinating. I don't have a good answer for that. The last two that I wanted to talk about relate less to location and more to what was happening with the fish itself. So magnesium has been called in other species of fish the chemical clock. And the reason is because it is highly associated with growth. So when fish are growing quickly, magnesium is higher, and when they're growing slowly, it goes down. And so in some species of fish, cod, mostly temperate to Arctic fish, you can really see as water warms and they start to grow, you can see a peak and then it goes down, and a peak, and you can actually age the fish, know exactly the number of years it has been alive based on the magnesium. It's really hard to know exactly what was going on here, but there are pretty significant peaks in magnesium. And there's this nice, interesting dip here that make it seem as though it's following a trend related to something. And we know that magnesium is strongly related to growth. And we know that Belize is tropical and the water is not changing temperature very much. So it's probably not tracing temperature as much as it's tracing growth. And I would be very interested to be able to figure out whether you could use this magnesium to understand growth, and whether this fish is kind of growing, pretty normal in here, and then all of a sudden there's a dip in growth. Why? Is that really what happened in the life of this fish? That's something that we can't prove, but it gives us this little bit of data that we can say, oh, that's really interesting. Let's go figure it out. That's the cool part of doing work like this. And in the same thing is going on in the embryo. There's all this movement. Could that give us some indication of what's going on with sawfish in the womb? That would be cool. We don't really know much about what happens to them, in the womb, but maybe. I don't know, but it's cool. And then manganese is another one, and this one I think is potentially very interesting. The data is very noisy, but manganese has a very interesting relationship in the water, in salt water. Manganese doesn't move into the water column in large abundance most of the time when the water has lots of oxygen in it. And that's a chemical reaction related to redox potential that I won't get into because it's extremely complicated. But most of the time, you don't see much manganese in the water. But when you do see manganese is in close proximity to places that are reducing environments, places where things are rotting, or there's not much oxygen. So in anoxic locations, you see higher manganese. Where in the life of a sawfish would you see low oxygen environments that might bring magnesium into the water column? Mangroves is a big one, and there is a study in one species of shark showing that proximity to mangroves was related to higher manganese in their vertebrae. So, and we know that mangroves are incredibly important for sawfish rearing. So is it possible that you could look at the manganese throughout the life of a sawfish in their tooth and know when they were in close proximity to mangroves? And if this fish was caught near shore, is it possible that, through its life, there's some peaks, a lot of noise, but it's pretty low. But towards the end of its life, maybe it was moving into a mangrove swamp. Maybe that was, this last third where the manganese is increasing. Is it possible that it was moving into mangroves or into some area where there was a lot of decomposition happening and there's a lot of manganese in the water? I don't know. It's a good question. Another one that is fascinating that I'd love to explore. And then the same is true of the embryo. There's variation in here, but not very much. It would be very interesting to know, what that really relates to, what it really means. And yeah, I guess that's the story that we were able to tell with this data. And what I'm fascinated to know is what else can we discover with more samples? It's going to be really cool when we can analyze more of these samples and really start to understand what's happening. And I came to Brazil in, I guess it was 2019, and met with Rafaela and with George and with especially Patricia Charvet. And we have managed to collect a large sample of fish. I think we have 131 samples now. Patricia had a large number of samples that she was able to provide us, and there's been several samplings of sawfish in São Luís, and there are some in Pará that Tommaso was able to claim for us. So we have this large sample set, and it gives us the opportunity to start looking at this chemistry and saying, what can we learn? In salmon, which I'm most familiar with, I've done the most work, this sort of chemistry has absolutely revolutionized the studies that we can do. We can track salmon down to 5 kilometers. We know exactly where they were during their life. It's unlikely that we'd be able to do that with sawfish. The ocean is a different animal, but it still provides us all of this information, and all of this information that we can't collect from fish in the wild. And then we know that there are a lot of these sawfish rostrum that are saved for various reasons in museums and in people's houses, and there's a huge storehouse of information about what sawfish populations were doing in the past and in the present, that we could learn a lot from these saved rostrum without ever having to chase down wild fish and risk harming them. This could be something that that doesn't have to harm fish that are alive now in order to understand a huge amount of information that we could use to conserve the species. I'm just going to make sure, yeah, I don't have anything else to show, so I think I'll stop my screen. But yeah, I think that this chemistry is very valuable and something that I'm really excited to start to learn about how it relates to sawfish ecology and how we can use that as a way to understand and conserve the species. So yeah, thank you for having me here to talk about it.
Thank you! É uma ferramenta que dá tanta informação, né? E de um material que está parado há muitos anos, né? Que a gente imaginava, pelo menos para mim, foi uma grande surpresa, uma grande novidade, porque a gente acha que tem que trabalhar com material fresco, né? Material novo, captura recente, para ter informações, né, das espécies, e essa é uma ferramenta muito legal, porque ela conta toda a história de vida do animal, né, com uma informação mínima, né, um pedacinho mínimo, na verdade.
Sim, um pouco de mágico, né?
Exatamente. E aí, como é uma espécie muito ameaçada, né, a gente, essas ferramentas minimamente invasivas, não invasivas, né, elas têm que ser mais assim, buscar, né, a gente tem que buscar usá-las porque a gente realmente não tem acesso, né, ao animal novo, né, a gente é uma espécie muito rara de ser encontrada e andar, acho que eu não sei, né, porque mesmo, e mais a quantidade que tu conseguiste, né, juntar junto com a Patrícia, com o professor Jorge aqui na região, 100 exemplares já, né, que vai ter como analisar. Dois teve essas informações, imagina como vai ser esse artigo bombástico aí quando ele vier com todas essas informações. A gente está vendo aqui as perguntas. Eu queria saber, eu queria que tu explicasse melhor para a gente como foi a validação desse método para o Sawfish, porque eu já tinha ouvido falar pelo menos com vértebras, né, de tubarão, por exemplo, mas aí como foi a validação para trabalhar com Sawfish?
Sim, acho que a validação é difícil. I'll speak in English. Validating the method is something that's going to be difficult and require a little bit of thought. There's some things that we know about the chemistry that would lead us to believe that there is very little difference between the chemistry we see in teeth and the chemistry we see in vertebrae. So especially strontium isotopes. There's a huge literature showing that strontium isotopes are what is in the environment is represented in an animal or a plant almost perfectly, and that is true across species from plants to terrestrial animals to aquatic animals. So we know that those numbers are probably something that don't need a huge amount of validation. On the other hand, the elemental ratios, the strontium and barium, and especially the manganese and magnesium that I showed, those can be very species specific. Strontium to calcium ratio and barium to calcium ratio are very tied to the water, but they can still be very specific to the species and to into what's going on inside the biology. And then the magnesium and manganese are very, very specific. I think that we need to be able to pair the analysis of teeth with the analysis of vertebrae, which is hard for sawfish because it's very hard to get your hands on samples. I know that we have a couple that we will be able to use, but yeah, that's hard. And then there's one other really critical piece to validating the method, and that is that sawfish use those teeth and they wear down over time. So even though they're getting bigger, they're still being worn, and they have very specific wear patterns on the teeth. So every time some of that tooth is worn off, that is information that's being lost. So we're not seeing from birth to death. We're seeing from where the tooth has stopped wearing to when the fish died. And understanding how fast they wear is going to be very important to understanding what we're seeing and how much time is represented in each tooth. So there's a lot to do, certainly.
Começou agora, na verdade, né? A descoberta. Agora com essas os novos exemplares que tem, né? Qual é a expectativa de resultado que tu acha que vem com a análise desse material completo?
Desculpa, meu vídeo acaba por um pouco. Fala de novo.
Eu perguntei sobre a tua expectativa de resultado para essas novas amostras que tu tens agora, esses 100 que tu conseguiste, né, recolher, para fazer, qual é o resultado que tu espera das análises futuras?
Não entendo.
Ai, deixa eu tentar de novo. O que tu espera depois das análises do material que vem, do resultado que tu acha que vai vir?
Sim. So what I think will happen in the future with this analysis? Yeah, I think it's going to be interesting. I mean, I have big hopes for what we can do with this technique and with this data. I'm looking forward to doing the analysis and publishing on it. And I really hope that what it tells us is, well, there's two things that I think would be very valuable to come from this. One is, what are the nursery areas that sawfish use? We don't know where those are. And it would be great to be able to use that to, you know, in other fish species, we can use the chemistry to find the location that a fish was. Even in the ocean, we've had some success finding the location. So I would really like to see this type of analysis allow us to locate nursery areas and rearing areas for these fish that we don't know currently, that need to be protected. And actually, I'm working with Ecologists Without Borders, a nonprofit in the United States. They have been doing GIS work to create predictive maps of the coast, the mouth of the Amazon and the Amazonian coast down to São Luís and up to Amapá, to understand salinity and temperature. We're hoping we can understand depth, because we know that sawfish don't move too far offshore, in the hope that we can use those maps and the chemistry to start to understand population structure in these fish. So that's another thing that I hope comes from this work.
Massa. Então, aí a utilização dessas informações poderia converter, por exemplo, em estratégias de conservação, de exclusão de pesca, por exemplo, áreas, né, mais restritas, unidades de conservação. O objetivo é mais ou menos por aí, né?
Sim.
Yes. Muito interessante, muito massa. O que mais? More questions? Rolou muito um vídeo por aqui de uma captura de um Pristis enrolado numa rede, e aí os pescadores arrancaram, né, a katana do animal ainda vivo, né? Tu teve acesso a esse vídeo, tu viu?
Sim.
E aí trabalho.
É muito triste. Sim.
Eu te confesso
Muito
que eu não
pior
consegui assistir o vídeo.
Sim. É muito triste que o vídeo ocorra 3 dias antes do International Sawfish Day, né? Horrível.
Exatamente. Então, e aí a gente vê então que ainda tem muitas dessas capturas aqui, né, nessa região amazônica. Aí fica o questionamento: será que aqui é um refúgio ainda para essas espécies de pristis? Porque no restante da costa do Brasil já deu uma desaparecida, né? Será que aqui seria um lugar potencial para encontrar essas áreas, como tu falaste, essas áreas de berçário? Tu acha que...
Yeah, this area, or the Amazon region, is such an important part of the, I mean, it's one of the few places in the world where sawfish are still hanging on, and it would be great to know more. And it would be great if there was the means to communicate the importance of sawfish in the region and really do more work about understanding where they are and what they're up to.
Tu acha que um trabalho com pescadores artesanais, conhecimento, resgatando deles o conhecimento que eles têm da região, tradicional, de captura, de pesca, tu acha que é uma estratégia? Com certeza eles têm mais contato do que a gente, né, com esses animais.
Sim.
Eu trabalhei um pouco com conhecimento tradicional, mas conciliar um trabalho tanto de resgate dessas informações que eles podem trazer, como de repente, sensibilização dos pescadores. Eu acho que poderia ter uma ação, né? Muita coisa legal para a espécie aqui na região costeira. A história vai investir um pouquinho por esse lado nos próximos. Any more questions? Let me see. A gente não tem mais perguntas. O que é bom dura pouco. A gente pode... Muito bom conversar contigo. Muito obrigada por ter hoje, né? comemorando, não deixar esse dia passar. Se tu quiseres passar alguma mensagem, falar alguma coisa a respeito da ação para a gente finalizar, fica à vontade.
Muito obrigado para sua invitação para falar um pouco. Muito obrigado.
Eu te agradeço, thank you. A gente encerra, então, aqui a todos que participaram. Não esqueçam de seguir o canal, dar o joinha no vídeo, curtir. E por hoje é isso.